She is a graduate student, more than a graduate student, you're a PhD candidate, at the University of California, Berkeley, conducting research on chemical ecology. Chemical ecology. I hope you tell us a little bit about what that is. Behavior and evolution of spiders. By investigating the use of chemicals, you may have to help me, chemicals in Tetragnatha.
Tetragnatha.
Okay. Spiders across Hawaii, California, the East Coast, Japan, and Germany. She hopes to find, to better understand how spiders produce and use chemicals and how that has shaped the evolutionary path of this lineage. So, Ashley, you are now ruler of the remote.
Thank you. Great. Well, hello, everyone. Thank you so much for having me tonight. I'm really excited to talk to you guys about spiders and geek out about kind of how chemicals and spiders interact. So hopefully I can get through as much as I've prepared, but we'll see how far I get. So first of all, as Ron introduced, my name is Seira Ashley Adams. I'm a graduate student at UC Berkeley studying spiders. So I'm part of the Berkeley Evo Lab working with Professor Rosemary Gillespie. Our lab is generally interested in understanding how biodiversity works and how evolution works. And I specifically kind of take a chemical ecology angle to it. So I'm interested in how chemicals interact or kind of relate to the evolutionary process that we see and the biodiversity that we see, and especially using spiders on islands in Hawaii as kind of my study organism. So hopefully by the end of this talk, I can convince you all that spiders are really exciting and our lab does some cool work. So if you are interested in seeing what we do have a lab Instagram, berkeley.evolab, where we post some unique kind of insider look on what a graduate student life is like or what a postdoc life is like. And then we also have a Twitter account that we post publications that come out from our lab, any kind of honorable mentions, cool studies that we find interesting. So please check that out if you're interested. So today we're talking about copper. And like Ron introduced, copper can be found in certain organisms. But for those of you just kind of stepping into the room now, what does copper have to do anything with spiders? Other than this wonderful copper wire spider art, the answer lies in, again, hemocyanin. But what is hemocyanin? And again, Ron talked about this a little bit, but I'm going to go over it again. Before we can really talk about hemocyanin, we need to talk about hemoglobin. So that's something we all have, and so hopefully we should be familiar with our little hemoglobins in our body. But hemoglobin is a respiratory protein that can be found in the blood cell. So it's a protein complex. It's made up of these different subunits, and as you can kind of see right there, in each subunit there's that iron compound that is basically the binding agent that binds to the oxygen, and then the hemoglobin can take it around throughout the body. So that's hemoglobin, but hemocyanin. So that's also a respiratory protein, but instead of using iron, it uses copper. So like hemoglobin, it's made out of different protein subunits, and if we kind of break down the units up, we get down to this main little subunit, and in the middle of the subunit, you see this active site. So the red is the oxygen, so O2, two oxygen, kind of combined, that's one oxygen molecule. And then the blue, or the kind of the purplish color, is copper. And so if you look at it, the chemical structure, this is what it looks like. This is actually the deoxyhemocyanin form, so this doesn't have any oxygen. So in order to get the oxygen, this is what happens. So just want to point out that's where the copper is. So you have two copper molecules. And so you have this oxygen kind of floating around, and then the copper binds to it, and then it becomes oxyhemocyanin. So that binding of the two copper elements to one oxygen molecules is what happens in the hemocyanin. And as you can see here, the hemocyanin protein can come in all different shapes and sizes. Basically, across the different organisms that have hemocyanin, it can come in big, bulky structures. It could be small, it could be narrow, all sorts of shapes and sizes. But who has hemocyanin? So Ron mentioned horseshoe crabs, but there's actually some other stuff. So this is a tree of life, kind of a little bit, trimmed a little bit. We as vertebrates are in the group Chordata. We're chordates. And our other vertebrate friends, we all have hemoglobin. But hemocyanin can be found in mollusks. Mollusks are squids and snails, but also hemocyanin can be found in arthropods. So arthropods are, of course, our lovely spiders as well as other organisms. So we're going to kind of look into arthropods a little bit more. So if you break down arthropods, you get these big groups. The chelicerates, or the chelicerata, this is where we find the horseshoe crabs and the spiders. Myriapods, we have millipedes, centipedes, crustaceans, crabs, and hexapods are insects. So all of these organisms have hemocyanin to a certain extent, except for hexapods, or insects. Basically, the vast majority of insects do not have hemocyanin. A few do. That's kind of through recent discovery we found that out. But I'll be talking a little bit more about why they might not have hemocyanin later. But our main focus here is spiders, so we're going to look at chelicerates. So the tree Chelicerata has a bunch of really unique organisms. Just to name a few, we have sea spiders. To be honest, when I first saw this, I thought it was like a piece of ginger that someone was like giving a joke, being like, hey, this is an actual living thing. It actually is a living organism. They are a very basal group in the chelicerates. They live in the sea spider. So they're a really unique group. These are the horseshoe crabs. We have Apilionids, which are harvestmen. They're also called daddy longlegs. You see them a lot in the Berkeley Hills. Sulfugids, sun spiders, camel spiders. We have, of course, scorpions. And then we have our spiders. So from the research that we have today, we know that these groups definitely have hemocyanin. And also from this kind of similar vein of research, we know that these groups don't have hemocyanin. So why do some groups have hemocyanin and some groups don't? In order to answer that, we're going to need to look a little bit closer at spiders and their respiratory system, because hemocyanin is a respiratory protein. So I want to talk to you a little bit about the whole system here. So when you look at a spider from kind of the sideways profile, this is generally what it looks like. You have your kind of the head area, which is the cephalothorax, and the body is the abdomen. So if you look at the inside, this, it's jam-packed with all these different internal organs. But we want to focus on the respiratory and circulatory system. And so it basically boils down to this. And if you look at it, you see this interesting structure here, and it's called the book lungs. Basically, the book lungs, it's situated at the base of the abdomen, or we call the ventral side, so kind of on the tummy side of the abdomen. And so if you look at a spider from top down, if ever you see a spider and you have the guts, and maybe the spider is knocked out or something, you can flip it over, and this is the book lungs. It comes in pairs. And this spider has actually 2 sets of book lungs. And so this is really where the book lungs are situated, right kind of under the cephalothorax on the abdomen. So when you look at each kind of lung piece, you'll see that at the bottom there's an opening. This is called the spiracle. It's basically just a giant opening that opens the whole spider up to the external world. That's where the air goes in to the book lungs. And if you look at a cross-section of the book lung, kind of like in this diagram here, you see that basically you have your, the S stands for the spiracle, and then the AT stands for the atrium. So this is where the air comes in, it flows in, and it goes down these kind of long channels. And because the channels are almost like a layered cake, but someone thought it looked like a book, kind of looked from the side, so that's why it's called the book lungs, just really from how it looks. So the air that comes from the spiracle into the atrium, down these different channels, the oxygen in the air then diffuses across from the open space into the spider's hemolymph, which is basically the body fluid of the spider. And in the hemolymph, you'll find hemocyanin. So the oxygen that comes in, dissolves, kind of goes across the membrane and into the hemolymph. The hemocyanin latches onto it and then transports it across the body of the spider. So now we know that spiders have book lungs, but spiders are so interesting in that they actually have another set of respiratory organs or systems over kind of on the bottom side of the abdomen called the trachea. So the trachea is, you can think of it this way, it's kind of like a series of pipes. So you have an opening at the base that opens up the spider to the whole entire world, and it's just a continuous branching line of pipes. And so this pipe, if you actually digest away the tissue around the trachea, this is what it looks like. So this is a part of a trachea from a spider. You can really see that it's just, you know, this one, there's a main trunk, and then you have all these different tubes and pipes just flowing out of it. So the air that comes from the outside can go through these pipes and through all to the different little parts and nooks and crannies of the spider. And the neat thing is that this trachea system is very different across the different families of spider or groups of spiders. So you can see that some spiders, it's really simple. You just maybe have a main tube and then a few branches. In other spiders, you actually have a lot of different branches, and we can call this like a really well-developed tracheal system. And the other neat thing is that actually insects use the trachea system as their main respiratory organ. So if you look at an insect, you can see that they also have a very well-developed tracheal system. So it's really innervated and it's really branched. So, copper. So now you know that spiders have book lungs and trachea. It's not like one is more important than the other. They actually work, they can work together. Some spiders, you know, they use the trachea more, they use the book lungs more. It's really that they are different, they do different things, but their main goal is, of course, to provide oxygen to the body of the spider. And so if you look at the tree of life of spiders, you can see at the base, this basal organism, what we call the mygalomorphs, they only have book lungs. So they actually have 2 pairs of book lungs. But if you look at a more derived kind of spiders, the araneomorphs, you can see that many of these araneomorphs actually have lost the second set of book lungs and have evolved that tracheal system. And another cool thing is that there's actually another spider in there in the araneomorphs, the pholcids, which I actually brought a little pholcid spider here. You'll find them all over your house most likely. I think they're adorable. But these guys, they actually have lost the tracheal system and only have 1 pair of book lungs. So it's really variable across the different spider groups. But you're probably thinking, well, what does this have to do with hemocyanin? The cool connection here is that hemocyanin is extremely kind of related to book lungs. Why? Well, because when you have the book lung, when the air goes into the book lung and diffuses across the membrane, it's not going to like make its way through into the brain or into the other vital organs. It might just sit there, or it might take forever to really get to the other parts. So you need a system or a way to get that oxygen to the other parts of the body. That's where hemocyanin comes in. They are the transport system. But on the other hand, the trachea, they have the transport system already built in to their whole structure, because they are basically like the train tracks that takes the oxygen all the way to the vital organs, their vital cells. So they do the work for themselves. That's why in the trachea system, the hemocyanin becomes kind of redundant or not as essential of a component. Whereas the book lungs, the hemocyanin is very essential. So this is where it gets really interesting. This needs a lot more research, but there's actually an interesting trend where when you look at the kind of the different tracheatedness or the differently developed tracheal system in the spiders, the more developed your tracheal system is, you see this trend where you have a lower concentration of hemocyanin in your body. And if you remember, insects have a tracheal system, and they also don't have hemocyanin. And just to kind of add, there's actually 1 genus in the whole entire spiders that we've actually discovered so far. This one genus, Disteridae, they don't have hemocyanin, and they also have this really well-developed tracheal system. So this might be kind of one reason as to why we're not seeing hemocyanin in certain organisms. Now, to wrap up this story, I kind of wanted to pose a question that, hopefully to get your thoughts going. Why don't we see ginormous tarantulas the size of buildings? Or in my, little vision, why don't we have little like cute puppy-sized tarantulas that you can cuddle up to? Why? Why don't they get that big? Well, one of the reasons that we think that might be is their respiratory system. So unlike our respiratory system, their respiratory system is actually very passive. So imagine this, when you're working out, when you're really exerting yourself, you need more oxygen. Your body's expending energy, your body just needs more oxygen. So to compensate for that, you breathe harder. So you can take in more oxygen actively by breathing and getting more O2 in there. But on the other hand, spiders and insects, their system works very passively in that they just have a hole. So whatever air is going in and out from that hole is the oxygen that they have to work with. So there might be an upper limit to how much oxygen their body can take up or use. So that might be what's kind of possibly what's keeping them at a certain threshold. And the other component is really the hemocyanin. Hemocyanin is regarded as less efficient compared to our hemoglobin at transporting oxygen and just taking in oxygen. So with the hemocyanin added on to this kind of limited respiratory system, in the current system that spiders have, it is most likely that we'll never see ginormous spiders, sadly, or maybe wonderfully, who knows. But interestingly, in the prehistoric era, when the oxygen content in the atmosphere was a lot higher than what we experience now, you did see big, not spiders, but big dragonflies and insects. So it seems like if there is a higher concentration, maybe they are able to kind of stretch that size boundary, because the bigger you are, the more oxygen you're going to need. So with that, since I have more time, since we're talking about copper and chemicals, I wanted to talk about chemical ecology, since that is kind of one of the main things that I do in my research. So chemical ecology is a field where we're trying to basically understand the chemical interactions that organisms have with their environment. It could be with other organisms, it could be with the abiotic environment, or like rocks and different cues in the air. So chemical ecology is a really interesting field for me, because we're interested in that interaction, but in a kind of evolutionary context. How does that chemical interaction or communication influence their organism's behavior or their ecology and eventually their evolutionary path? Chemical communication or the use of chemicals is very ubiquitous across the tree of life. Bacteria use it, we actually use it, and of course spiders use it. So a lot of different organisms use chemicals, and it's so versatile. So some of the ways that spiders can use chemicals is to use it to detect prey, to lure prey, to attract mates, to assess the environment, and to avoid predation. But this is actually only the beginning. Chemical ecology work in spiders is not a new field, but it's a relatively untouched field. So there could be still a lot more that spiders do with chemicals that we don't know of. So it's a really great kind of field to be in. But before we can talk about how spiders do these kinds of things, we need to talk about, how do spiders sense chemicals? Do they have a nose? Do they taste it? How do they do it? So the answer is actually they have hairs. We call this chemosensory hairs, but if you look at part of their leg, kind of really zoom in, you'll see, one, that spiders are super hairy, but two, kind of where the arrows are pointing, you'll see that they have these interestingly shaped hairs. And these are the chemosensory hairs. So we call them S-shaped hairs, since they have a kind of S-shaped curviness to them. But if you look at the tip of the hair, there's an opening. So insects also have this kind of chemosensory hair. We call them tip-pore sensilla. But this, we strongly believe, is the way that spiders are sensing chemicals in their environment. Unfortunately, this is something that we're still really working on. A colleague of mine is trying to figure out the ultrastructure of the hair, so cutting it up into slices, looking at whether it's innervated, how many nerves are going through it. All of this we're trying to actually figure out right now. And one of the work that they have produced in their lab is this great mapping. So she took the time to take all the legs of the spider and map out exactly where the chemosensory hairs are found on a spider's leg. So this is leg one, so the first leg of a female. It's broken up into two parts. Part one is kind of closer to the body. Part two is the more distal end. And really the take-home message here is that the chemosensory hairs are all over. They're not just situated at the tip where maybe the spider touches things, but they're up here where we don't think they're going to be touching anything unless they're brushing up against someone. But they're basically all over the place, and you do see an interesting pattern where they might be highly, they're denser at the tips. So really this is kind of a step into better understanding how spiders sense the chemicals. And another thing I kind of just want to quickly touch up on is that in the chemical ecology world, we can kind of, even though it's a spectrum, we can divide the chemicals into two big categories. One is the olfactory compounds. That means these compounds are usually lighter and they're a lot more volatile. So organisms sense it in the air. They kind of smell it. That's kind of what we're talking about here. So an example would be a female moth producing sex pheromones that the males are, as they fly, cueing in on and using to guide themselves to the female. Another group of chemicals we can call are the gustatory compounds. So gustatory chemicals are the opposite in that they're heavier, they're maybe waxier, they don't really have a huge volatility or high volatility, so they stick on the surface. So here a fly is maybe tasting the wax or the sugar in the fruit. That would be kind of a gustatory compound. In insects, we know that there are actually two different chemosensory hairs that detect these two different compounds. In spiders, we only have one type of hair that we've discovered so far, but we are pretty sure from behavioral evidence that they can sense both olfactory and gustatory compounds using this hair. So I'll get into some of the examples of how they use chemicals. So detecting prey. This is a story from a black widow spider, so Latrodectus. So scientists actually took some rocks and they had a cricket just walk all over it, live on it for a little bit. They took the crickets off and then they gave the spider the choice of that rock versus a control rock or a rock that had no cricket. And this is so there's no cue other than that kind of chemosensory cue that the crickets left on the rock. And we see that the spiders prefer the rock where the cricket or the prey had been on. So here's an example where they actually can use chemosensory cues or chemical cues to really choose an environment to maybe hunt or increase their efficiency of hunting. Another kind of detecting prey story that I want to talk about is about these wandering spiders, Cupienius. But before I get into that, I'm going to diverge a little bit and talk about the venom gland. So venom. Spiders use venom to subdue their prey. And venom is actually a really interesting chemical cocktail. So we call this venom cocktail when all these different compounds are mixed in, really like a cocktail drink. Maybe someone, is there like a venom cocktail? That'd be really cool. But it's basically like a cocktail drink where you mix all these different components. Some spiders can have up to 1,000 different chemicals mixed into their venom. So a venom is very rich. And each different spider has a different mixture of chemical compounds in their venom. And the cool thing is that the chemical mixture can have different effects on different prey or on different organisms. So an example of this is so this is from a group of venom compounds called latrotoxins. You can find them in black widow spiders, but you can also find some latrotoxins in the common house spider. The difference, even though they have different latrotoxins, the really unique difference is that in black widow spiders, they have this latrotoxin called alpha latrotoxin, and that toxin specifically targets vertebrate nerve cells, and it doesn't affect invertebrate nerve cells. You'd think that's weird because spiders eat insects, right? Well, the idea here is that the black widow spider builds webs that sometimes entangle vertebrate prey, like little lizards and little mice. And so through evolution, they've developed this latrotoxin that targets vertebrates specifically, whereas the common house spider, they don't have any of that compound. So that's why when you're bit by a black widow versus a common house spider, as a vertebrate, it hurts a lot more with the black widow since they have this alpha latrotoxin. So knowing that, we're going to go back to the wandering spider story. In the wandering spider, that spider's venom is actually very potent against crickets. So a small dose and the cricket's pretty much a goner. Whereas in cockroaches, they're not very efficient. So you'd either have to inject a lot more toxin or you'd have to wait longer until the cockroaches hopefully eventually is subdued. So knowing this, scientists did this really interesting experiment where they took filter paper and had crickets run across it. They took another filter paper and had roaches run across it. And then they gave the choice, just the filter paper, to the spider and looked at what was his or her preference. Now, a wandering spider on a full venom gland, when they have all the venom that they can have, they preferred both prey items. But when the spider had a nearly empty or empty venom glands, which actually the scientists milked you can milk them by kind of sending an electric pulse that makes the muscles kind of contract and then pushes the venom out. So that's how you can milk the spider venom. So they milk these spiders, and so in a nearly empty venom gland, the spiders actually preferred the crickets. So not only was the spider able to distinguish cricket scent from cockroach scent, it actually affected their behavior. And so this is a kind of, I thought, a really unique system. And then lastly, for the detecting prey, I think this is a really cute story about vampire spiders. So this is a type of jumping spider. They actually hunt mosquitoes. And so scientists gave them the choice between, this is all chemical, none of the organism, just the chemical cues of a normal, kind of hungry mosquito versus a blood-fed mosquito, and looked at whether the spider had a preference, a chemical preference. And they found that these vampire spiders actually preferred a blood-fed mosquito. And the cool part of the story is that actually the opposite sex, so both males and females, prefer mates that have fed on a blood-fed mosquito. So you have this kind of a whole circle here of chemical cues. And these mates actually find know that the other spider have eaten a blood-fed mosquito just on chemical cues alone. So this is kind of, it goes into attracting mates and other stuff. So speaking of attracting mates, we actually don't know too many compounds, actual compounds of a spider, but some of the compounds that we do know come from sexual pheromones. So in the wasp spider, Argiope, you should find them in Berkeley too, around the Bay Area. Argiope spiders, they have this kind of sexual compound that females emit called trimethyl citrate. So adult females, virgin females, produce this, and males are attracted to this. So the researchers is really cool. This is actually a colleague of ours. She was able to get this compound synthesized, so created, and then she took that synthesized compound, put it on some stuff like cotton balls, and then put it out in the field. And then within minutes, there was just a horde of males running toward that synthetic compound and like doing little sexual behaviors on it. So really, we know that this is very potent. And the cool thing is that in juveniles, they don't produce it, of course, that makes sense. But in mated females, once they mate with a male, they stop producing it as well. And on the flip side, kind of keeping that in mind, the fulcid spider, so these guys you'll find in your house, and I brought one, a male, along. So the fulcid spiders also have a sexual compound that the females emit, that again, the males are attracted to. But when these spiders mate, the females continue to produce this compound. The only time she stops producing, or rather she decreases the amount of this compound, is when she has an egg sac in her mouth. So fulcids are really cool in that once they lay an egg sac, rather than abandoning it and leaving, she actually carries it in her mouth. So when she's carrying these egg sacs in her mouth, she doesn't produce, or she lowers the production of these sexual pheromones. And then kind of going into luring mates, I wanted to talk about luring prey. This is for me I would love to one day see a bolus, one of these spiders up close. This is one of my favorite stories. So the bolus spider, they are in the genus Mastophora. So the bolus spiders, they actually can lure prey using chemical cues. So, but before that, they don't make webs. They actually make this kind of silky material structure called the bolus. And when you look at the internal structure of a bolus, you can see that it's just a twine of all these silk kind of mixture, or, well, they basically kind of scrunch up all the silk into a ball and then coat it with this liquid gluey material. And these spiders can actually produce the scent of a female moth from their body. And that's what they use to attract the male moths to them, and then they whack the moths with the bolus that sticks to the moth, and then they can eat it. So I have a little video here. So that's the female making the bolus. You can see she's kind of pulling out the silk from her spinnerets, and then eventually she will finish the bolus. That's her finishing it up. And then she's gonna climb up, take the bolus, and what they do is they basically just sit there. These guys are nocturnal, so they hunt nocturnal moths, mostly in the noctuid family, I believe. And so they sit there, and rather than the ball producing the pheromone, it's actually their body. Their body produces, and it actually perfectly mimics the female moth pheromone. And so In this video, they actually tethered a male and was like to the spider. But in the wild, the male would be attracted to the scent, thinking it's a female, go up, and the spider will just whack it with this bolus, catch it, and then inject venom into the moth. So this is kind of, I think, a really neat story. And the really neat thing about them is that there's different species of bolus spiders, but some bolus spiders can produce chemicals that perfectly mimic a specific species of moths. And so how that happened evolutionarily, we don't know, and that's actually another kind of interesting question we're trying to answer. So with that, I hope that I was able to kind of convince you that spiders are really interesting creatures. There's a lot more to spiders than just this, but hopefully you were able to understand why copper is important in spiders. And in the future, I hope to see you guys more at these talks. So thank you so much for having me.